So here's my favorite part of the tour, sitting down. Yeah! Favorite activity. I'm gonna hit this.
Okay, don't mess with that thing now.
Okay, does anybody know what this is? Coral. Does anybody know what kind of coral this is? Branching coral, very good. Does anybody know what kind of branching coral it is? Brown branching coral? It's good that you're using your observation skills. It's called Staghorn Coral because it kind of looks like the horn on a stag. You see that? Alright. Yes. It's a very cool type of coral that I've actually worked with in other capacities. I've actually worked scrubbing these on the PVC little Christmas trees. It's actually really cool. These used to be one of the #1 corals out on the reef. Has anybody seen a coral that looks like this while they're down here? No, that's because they're probably one of, on this trip, you saw one of these?
No.
Oh. Yeah, I was like, please tell me where. They used to be, yeah, like I said, one of the #1 corals on the reefs out here, and now they're all but gone. So that's like if the most numerous species of tree in the rainforest disappeared. There would still be forest, but it would be a lot less dense, and there'd be a lot less habitat for the things that live there. So they're trying their best to regrow them. So the scientist in charge of that program is called Eric Bartels. They focus on Staghorn Coral, like I said, but they also focus on Elkhorn Coral. They're very similar. They're in the same genus. Did anybody see Elkhorn Coral out at Lou? I know I did. You did?
Yeah.
Yeah, it went out and it was really big and orange, and then it sort of flattened out at the top. Yeah, so it's got horns that look a little bit more like elk, right? But they're very similar. They're very fast growing. They use these PVC, they call them trees, to grow them out, and they'll anchor these to the bottom of the ocean with a little ball floating at the top. And what that does is these guys grow really, really fast, and they grow out in all directions. So if you just attach them with some filament here, they'll just spike out, and then you can take them and you can adhere them to the substrate, and by substrate again I mean the bottom. You can drive a nail in if it's rock, and then zip tie it to the rock, and then maybe epoxy it down to the bottom, and it will just start growing from that point, if it is, healthy. So it's actually a very successful program. However, the downside is if you do that with a coral and you're just using more genotypes that previously died, that coral as well might die. So there's something a little more we have to do to help treat these guys. Just some facts on this. When these colonies reach 2 inches, they are ready to be outplanted. So that's even a little big for outplanting size. Yep. And there are many of these nurseries throughout the Keys. So this is a pretty widespread form of coral restoration. It's actually been around a lot longer than the type that sea base is doing. Sea base, by contrast to branching coral, we are working on regrowing bouldering or encrusting coral. So do you guys know the difference? Can you give me an example of boulder coral? You've answered a lot. I have faith that you know.
I can't give an example of it. It's like brain coral.
Oh, that's a perfect example. That's what I was thinking, because brain coral is my favorite kind of coral. I think it's super cool. There are actually 28 species in total of coral down here, and most of them are bouldering corals. So if we were to only save the branching coral, we'd only be getting part of the picture. Massive corals like brain, boulder, and star corals are the signature reef-building corals of this reef. So they're the main reef builders. Can anybody explain to me what that means? Oh, green shirt.
Does it mean like the foundation of like the whole ecosystem?
In a way, yes. So they're the one creating the most calcium carbonate. They're depositing the most physical reef structure down, right? These guys, they do have a calcium carbonate skeleton, so they are stony corals, but they're not going to create those huge spurs and grooves that you see at Lukey. All that physical habitat, that's mostly going to be those bouldering corals, so they're especially important. So just a little history on just the fragmentation process. Reskinning. Oh, okay. They're going to talk about that before they talk about the process. Okay, so what they do here, and what you'll see later today, hopefully, at Fort Sac, is they do a process called reskinning. So, I don't know if you guys see photos of it. There's some behind you. See all those little circles on that poster? Those little circles with coral? I'll draw it on the board, don't worry, I'm a super good artist. Alright, you'll take one piece of coral that maybe grows on the side of the ship. So, it's fair game for scientists to use and abuse. We'll take that one piece, and we'll break it up into a bunch of little pieces through a process called microfragmentation. A bunch of little pieces, but they're all from the same individual, so they all have the same genotype, right? You know what that word means? Right. It's like that, it's your unique genetic makeup, yeah. So, look, they have little pores, they're happy. Yay! Alright. And what they're going to do is they're going to take those corals and they're going to grow them to a suitable size so that they're healthy enough to be put out into the wild. That suitable size is generally about this big. Alright? And then they're going to take those and they're going to glue them to, let's say, a dead coral or just a piece of substrate, limestone out there. And they'll glue them on in little combinations. They've tried spirals, they've tried polka dots, they've tried squares, they try all sorts of things because science is about throwing stuff at the wall and seeing what sticks. Alright? They glue them to rocks down there. Now, if these are all the same genotype, if these are all genetically the same animal, what do you think those pieces are going to do? I'll let you answer.
Fuse.
Exactly, they're going to fuse together. So, they said, oh, this is all the same tissue. We're going to grow really, really fast. They're going to fuse. I don't know if you guys are following me, but you see that? They all fuse together. And then you went from small little things like this to something like this. And maybe I should redraw this picture because it looks like the before and after are the same size. Is there a, no, it's all right. All right. So you took a little piece like this. That's better. All right. So what makes the macrofragmentation process so special is that you can increase the growth rate of these really slow growing corals by up to like 40%, or sorry, 40 times faster. Thank you. So here's some stats for you. Should I have them? They did. I saw them. Okay, here we go. So, yeah, they say here, this is very impressive, they can create a coral in a matter of 5 years, so this process, end of 5 years, that would normally in the wild take 25 to 100 years to grow.
Wow.
Whoa.
Yeah. So,
Wow.
that is fantastic news, because we're definitely losing coral faster than the rate at which they grow. So, yes, evolution is always taking place, natural selection, all of that, but it takes place at a very, very slow rate, right? Humans didn't evolve in 10 years. In the same way, corals cannot adapt to the rate at climate change, the rate at which climate change is proceeding in a time frame where they'll actually be able to survive. So if we can accelerate their growth and accelerate their evolution, we may be able to save them. And this has actually been done to great success. And one of the places that you'll see one of these artificial reefs that they've made is Fort Zachary Taylor Beach, where we're going today. So that's pretty cool. You guys are seeing a little bit of good news in a sea of kind of dark news. So I think that's very special. What else do they want me to talk about? Okay. Yes. Now, this here, is this sexual or asexual reproduction? Asexual, right? There's no gametes involved, there's no sperms, there's no eggs, there's no recombination of genes. This is simply making one animal grow a whole lot faster to compensate for things like bleaching and disease. They also do manipulation of sexual reproduction of corals to bioengineer sturdier, more resilient genotypes and species of corals. And we'll see that when we move on to the next lab, which is right now. All right.